Good evening, sir. You can start now, sir. Good evening, sir. Sir, are you there?
Good evening, ma'am.
Good evening, sir. You can
Good
start
evening.
now.
Yes, ma'am. Thank you so much. Ma'am, please post disable participant screen sharing, ma'am. Excuse me. Yes, thank you. Good evening, my dear students. You are all are warm welcome to the new chapter. Strategy for enhancement in food production. Okay. In this session, what we are going to discuss, see that the plant breeding, okay, and also the development of the new varieties and creations of genetic variations, okay, and what are the subgetting is that, see here, and domestication, germ plasm collection, plant introduction, hybridization, polyploidy, mutation, and genetic engineering. So already all these things are related to the chromosomal abnormalities. Already we studied in so many times, isn't it, my dear? Structure of chromosomes, and also that the types of chromosomes, and also where there is a mutation occur, point mutation, inversion, deletion, addition, all those things already we discussed, isn't it, my dear? How it will be implemented into the food production, that things we are going to discuss in the chapter very deeply. So many times they were asked the question from this chapter, mostly 4 to 5 questions they were asked. Idea. Okay, my dear. So before that, we should know what is mean by plant breeding. It's the first heading of this chapter. Already you know, but breeding is nothing but the development, isn't it? According to the what is that? So many methodologically they were used to increase the yield. Okay, that is very, very important for the plant breeding. Likewise, the animal breeding is also there. Animal breeding means you know about that. Suppose if it is take an example, so white tiger is only one in the world, means you can create that one into up to 100 or 1,000 like that. You can produce more than thousands. Suppose those who are all are endangered species, exotic nature. Okay, that things we can create. Take an examples of dinosaurs is also one of them. What is that? Completely nil in the world, isn't it, my dear? But we can create that one. Suppose if you find out the bone marrow of the animals, we can create the new dinosaurs. Okay, likewise, the plant breeding is the first heading, my dear students. So here what we are going to discuss here, the plant breeding is the art and science of changing the genetic of plant for the benefits of mankind only. Okay, mankind only. What is the main purpose of this one? Mankind. Why it is used? Why it is used? First of all, you should know. Okay. Suppose during the time of 1951 to 56, 61 to 60, there is a great explosion is there. Okay. That time what happened, the population is also increased, but the lack of, what is that, food product. That time the scientists also, what is that, they did some research on the plant, particularly Sonara Sadbadi is one of the wheat. Okay, who is the father of green revolutions in India? M.S. Swaminathan. Worldwide, Norman Borlaug. Isn't it? These are the two scientists there. Worldwide, what is that? Important one. According to the plant breeding and also invention of the new variety of the paddy. Okay, wheat and so on. So is there. They will fulfill the entire satisfaction of the people, as well as the green revolution also done by the scientists. Okay, media. So the plant breeding is the art of science of changing the genetic of plant. Genetic of the plant means taken examples. Rice, isn't it? So 18 chromosome is there, right? Okay. 1 acre. Okay, 1 acre. Okay, how much they will produce? 100 bags. Suppose if it is the hybridization plant breeding varieties, the new varieties were discovered, so that gives which one, 1 acre, how much, 1,000 bags or 500 bags. So it is a, what is that? Fulfillment of the research. Okay, medias. This is very, very important. Genetic of the plant for the benefit of mankind only used for the social purpose and also man origin. Plant breeding has been practiced for thousands of years. That is very, very important. Not only it will get the first step, you will not get the succeed. You will not get succeed. And practice in more than thousands of years. Since the beginning of human civilization, take an example. Civilization, maximization of the food production involved. That is very, very important. My ultimate aim is, what is that? The maximizations. Already I told you here. Yeah, how much is that? 100 bags. Suppose if it is produced 1 acre, 100 bags means it may increase up to 1,000 bags per acre. Okay, that is our goal. That is the maximizations. Okay, not only that, the quality of the food is also similar. Okay, the yield, it will be very high in nature due to the plant breeding practice. Okay, media. So in these categories, what are the steps involved? What are the types of involvements is there? The food production, particularly the food productions, because food production is very, very important manually. Okay. So the first one is called the improvement of the crop genotype. Genotype means genetically we have to change the plant. That's why they have to genetic plants, which is changing the genetics of the plant. Genetics means what is that? Gene. Gene means what is that? Chromosome. Okay, my dear. Chromosome changes. Chromosome alteration also there. For example, the plant is very height. Okay. Okay, rice plant is also very height in Thailand. Taken in our area, that is maximum 2 to 3 feet only. So this is the tall rice plant. Okay, rice plant is there. Okay, in our cases, in our area, India, how much is there? India, there is a semi-dwarf varieties. What discovered by the name of the scientist called us? What is that? M.S. Swaminathan. M.S. Swaminathan Foundation, M.S. Swaminathan Laboratory also present in where in Goa. Okay. See that one. So the first one is also genotype. First of all, is noted down which one the improvement of crop genotype. and improved of animal genotype. Sometimes it may be animals it related to which one, the meat. Take an examples of meat. Okay, weight, take an example, and also milk. Okay, and also egg, for example, in the poultry house. Improvement of the environment under which the crop and the animals are raised. That is very, very important. These are the three involvement. First of all, you should change the genotype of a plant. This is crop is nothing but a plant. Okay, plant genotype. Suppose if it is animal genotype, animal is also very, very important because both are interrelated only. Okay, without animal, without plant, there is no animals like that, because we are all depends upon the autotrophs of which one in a plant. Improvement of the environment under which the crop and animals are raised. It is also very, very important. Suppose some of the plant, they will grow in the dwarf condition, isn't it? Dwarf. Dwarf. Drought. D-R-O-U-G-H-T. Drought in nature. Dry conditions. It may be changed into the, which one, moisture. Okay, my dear. So these are the genetically modification we can do in the plant breeding methodology, my dears. First of all, you should note it down. The plant breeding is the art of science of the changing of genetic of plants for the benefits of mankind. Plant breeding has been practiced for thousands of years. Since the beginning of the human civilization, the maximization of the food production involves the improvement in crop genotype, and second one is the improvement of the animal genotype. Third one is improvement of the environmental under which the crop and animals are raised. These are the food production involves. So go to the next one is the development of the new variety. How it will be developed, how it will be possible. Already you studied in the genetic engineering, biotechnology, and also chromosomal aberration, aneuploidy, polyploidy, all those things already studied, isn't it, my dear student? How it will be possible? See here, development of the new variety, how it will be. For example, IR8, IR8. Okay, and some of them also. Okay, see here. First, development of the new variety consists of the following main steps. First of all, you should note it down. What are the steps involved in the, what is the new variety development process? New variety. See, first, creation of genetic variation. Creation of genetic variation is the first step. And second one is selection. Third one is evaluation and release as a variety. Fourth one is seed multiplication and distribution of the farmer. These are the four important steps they were used for the development of the new variety. Genetic variation, what is the meaning of genetic variation? Already, Mendel did how many characters? 37 characters is there. This is 34 character is there. Among this, how much they were selected? 7 only they selected, isn't it? Height. Okay, simply we may call as height. Tall and tall, dwarf and dwarf. Okay, yellow wrinkled, green round. Okay, all those things. Position of the flower and also terminal, likewise, isn't it? These are the characteristics. Genetically variation means tall is the one of the character. Take an example, tall, dwarf. Okay, and selection. So you have to select this one. The same species also very, very important. Suppose same variety. Okay, same species. For example, cat plus cat, isn't it? Lion. Suppose they may be crossed with these things, animals. Suppose we are crossing like this, you will get new variety. Okay, my dear. So these are the selection process. Which animals, which plants we are going to select for the development of new varieties. That things is very, very important. So both are same species or same family. For example, potato. Potato is the Solanaceae family, isn't it? Tomato, it is also Solan families. Suppose we will cross these two things under the process of protoplasmic fusion. One of the process already we did. Protoplasmic Plasmic fuser. Okay, media. Protoplasmic fusion. As a result, what we will get in a new variety, Pomoto. Pomoto is the new variety. So these two are crossed. Hybridization takes place. As a result, you will get the new variety. So here, what happened on the upper part, you will get tomato. The lower underground stem modification is also potato. So you will get the two yield in a single plant. That is very, very important. This is a new variety. Okay, and also fulfill the human being as well as the society also, public. Okay. The evaluation and release as a variety. So this is the call. So sometimes they may give you a name. It's called as a ER8. So it's a variety, isn't it? 8. Likewise, there are so many names there. Okay. 20. Likewise, different name is there. After the completion of this work, we may, what happened? Recommend to the farmer. That is very, very important. So this variety it will get, you will get this much. The value of value is also very high. Like that, they have to, what is that? Marketing. Seed multiplication and distribution to the farmer. After that completion of work, we may move on to approach the farmers. Okay. See that first one, domestication, germplasm, collection, plant introduction, hybridization. These are the things. So creation of genetic variation. How it will be get, these are the methods. The domestication, germplasm is also one of germs means nothing but the, what is a microorganism, plasms, collection, and plant introductions, hybridization. These are the technique. We will get the new one. Then next, on the other hand, polyploidy, mutations, genetic engineering. Polyploidy already you know, but addition of things. For example, 2n plus n. So you will get 3n. Triploid in nature. Mutation means already in our, but there is a chromosome is there. Take an examples: adenine, guanine, cytosine, thymine, like that. Isn't it? We can replace it. First of all, the adenine replaced by G means the total variation is there. So that is called as mutation. It is due to the physical and chemical biological process which occurs in the chromosomal structure. So it is also one of the mutation. It may be artificial or natural. Genetic engineering, man-made selection of process. Already we discussed genetic engineering, different classes, different plants, different animals. We can select the particular gene for the responsible for the production of the whatever it may be, antibiotics, insulin, hormone, whatever it may be. Okay, you have these things all essential for the process of genetic variation, creations of genetic variations. Then selection procedure, which one, which A or B sample, which gene, and the evaluation and the release as a variety. So after the completion of the hybridization, practice it so many times within the lab. After that, when it succeeds, so it can be evaluated. Then what is that? And the distributions. The seeds are, what is that? Seeds are, what is that? Multiplication. Particular seeds, I discovered one. For example, MS 9023. Likewise, imagine. So this variety I introduced myself like that. After that, it may be multiplied into millions, isn't it? Millions. Then only we can move on to the farmer. Okay, my dears. That is very, very important. These are the things is very, very important. Okay, my dear. Go to the next one. See that one? The creation of genetic variation. The genetic variation provide the raw materials for the selections, whereas selection affect the desired improvement. This can be done in several ways. Okay. Selection is very, very important criteria in this category. Genetic variation, how it will be? Genetic variation, which plant, which animals we are going to create the new variety. That things you must select. Okay. Selection is a process, first one. Then only you can improve this, can be done in several ways. How it will be? What are the methods that were used for the genetic variations? See that? Domestication, germplasm collection, plant introduction, hybridization, polyploidy. Okay, media. So next, see, mutation, genetic engineering. These are the, what is that? Selection process is there. So then only the process, it will be high. Finished. Okay. First of all, domestication. What is made domestication? Okay, here. It is the process of bringing a species under human management. Domestication means under the human management. The domestication of the plant is an artificial selection process conducted by humans to produce plants that have more desirable traits than wild plants. The practice is estimated to date back to 9,000 to 11,000 years. How many years they are practicing for each and everything? So, for example, take an example, say any one of the plant, take an example, rice, isn't it? Is that normal one? The normal rice, how it will be converted into genetically GMO, genetically modified organisms, plant or animal, whatever it may be, how it will be changed. So the normal rice chromosome is, you have to take and you have to identify the chromosome structures and select the length of the plant or content of the gene, whatever it may be, the molecules. You have to remove it and add the new one. That one also you have to select from the B sample, A sample. This one is B sample. You have to select the chromosome here, O. So replaced here. So it is also grown into the where the plasmid as a vector, plasmids as a vector. After that, what multiplication takes place within the microorganism, then it introduced into the plant. After that, you will harvest so many years, practice it so many times. First time it will not get success. Human error is also done. Okay, practice it more than 100 times. Then only you will get the good result. That is the reason. And many crops in present days, cultivations are the result of domestication in ancient times only. That is very, very important. Practice it. Which one? Ancient times. How many times? About 5,000 years ago. Nowadays, we are using so many crops, isn't it? These crops and the crops also practice it how many years in the 5,000 years ago in the old world and 3,000 years ago in the new world. Okay. 3,000 years. This is very, very important. Today, all of our principal food crops come from the domesticated varieties. Now we are using, what are the things, whatever it may be. Take an example of basmati. Isn't it, my dear? It's one of the new variety. Recent year only we are using this one. But this varieties also, they were practiced so many times in the domestication laboratory aspects. Then only it will be introduced into the market. Okay, my dear, this is very, very important. The next, the plant whose origin or selection is primarily due to the international human activity is called as cultigens. See this one? International human activity is called cultigens. And the cultivated crab species that has evolved from the wild population due to the selective process from traditional farmers is called as, what is that? Land raise. Land raise. That is very, very important. So two things you should note it down here. So one is called as, what is that, cultigen. Cultigen is nothing but the cultivated crops, already cultivated. How many years? Thousands years or 5,000 years. New varieties also, the old world. And also, how much years? 5,000 years. New world. So 3,000 years. The methodology is called as, what is that? Cultigens. What is the meaning of cultigen? Primarily due to the international human activities only. Ancient ages. How many years before? 5,000 years before. What they did. They practice so many times the domestication aspects. Domestication means, what is that? They prepare only themselves. Practice it so many times. Then only they will get the good result. After that, introduce into the farmer. That is called as land raise. Okay, my dears. Go to the next one. And second process called as germ blossom theory. Germ blossom collection. Imagine, this is also second types. Germplasm is the sum total of all the alleles of the genes present in a crab and its related species. Present in the crabs, alleles or genes. Thus, the germplasm of any crab species would consist of the following types of materials. Okay. Germs plasm means it is nothing but the germs is nothing but the, what is that, germs is nothing but the alleles or genes. Okay. So selection is very, very important. Germplasm. Here, what are the cultivated improved variety? Improved variety that are no more in cultivation. Old local or daisy variety. Lions produced by the plant breeders. Wild species related to the crab species. These are the, what is that? Material is also important. Following types of material is very, very cultivated, improved variety. Cultivated. So, for example, take an examples, MS20. Okay, just imagine. This variety is cultivated how many years before? 1000 years before. 1000 years before they cultivated. Now, what happened, the MS20 is also, what is that? Improved 20 into 30, 40, 50, 60, 70, like that. You can imagine, whatever it may be. These all are, what is that, new variety in the new world. That is called as cultivated improved variety. These are the improved variety. This one is the old one. Okay, these are the improved one. Improved variety that are no more in cultivation. And further, what happened now? The no more cultivation is there. Further, it will not cultivated. Old local or daisy varieties also. This one, old variety and daisy variety are called as old variety. Old means this one. Old means this one. Other name is called as daisy variety. Lions produced by the plant breeder. Lions, what is the meaning of lions? These are the new varieties produced by the plant breeders. Wild species related to the crab species. Wild species and also a crab species. These are the two things is very, very important. Okay. A good germplasm collection is the essential for the successful breeding program. You have to select the variety. You cannot select the bad quality or bad seeds. The old variety have now been mostly replaced by the more uniform new variety. That's why I told you old variety is replaced by the new variety due to the, what is that? Uniform new variety. In addition, say expansion of the agriculture, industries, and other human activities are damaging the wild relatives of the craft plantax. All these factors are causing loss of genetic variability and this germplasm. Suppose the old variety is there, okay, right, okay. The old variety is also replaced by the new variety, okay. So old variety is completely, what happened? Genetic variability, that is loss of, this one is called as loss of genetic variety. This one is deleted, isn't it? Old is deleted. But new one means, what does that mean? We discovered new varieties. The old one is completely, what is it? Genetically lost. That is very right. Germplasm is collected from within the country as well as from the other countries also. They may collect the plant, for example, rice, okay, sugar cane, chili, okay, my dear. So these are the plants we can collect from the same country. For example, Tamil Nadu, Andhra, AP. Or for example, Tamil Nadu, Kerala. Okay, my dear, like that. We may collect. Suppose if it is not possible, India and what is that? India and states, USA, India and Australia, India and New Zealand. Okay, New Zealand. So likewise, we may select the different countries. Okay, see, this is the first thing. The germplasm collection are usually maintained at a low temperature in the form of seeds. That is very, very important. We can store after the collection, we have to store in it. How much? You know about that cryopreservations minus 196 degrees Celsius liquid. What is that? Liquid nitrogen. Already we studied. This is very, very important, my dear student. Nitrogen. So many times they ask the question in this post. Cryopreservation. For example, egg, spam, as well as the seeds also, they can store in thousands of years under this temperature. Minus 196 degrees Celsius. Liquid nitrogen, don't forget, my dear student. This is very, very important. NEET point of view. So many times they ask the question, NEET point of view. Okay. And the next one is that the stored seeds are grown periodically in the field of obtained fresh seeds. This becomes necessary because the percentage of seed germination decreases with the storage time. This is also very, very important. For example, during the time of cultivations, maximum 100% seed germination is there. Okay, in the lab, just imagine in lab. Suppose if it is cultivated in the normal land, what happens? You know, they can grow normal land. Only 50% only means 50% will be lost, like that, isn't it? So when it attained 100%, it is a good result. Okay, so you have to practice more than 100 times. Then only the seed will be selected and collected and also introduced into the farmer or approach to the farmer. For example, take an example. Take an example. A particular seed is there. Just imagine this is the first time. Okay, the particular seed is there. They will grow under the field. Okay, they grow under the field. So how much they got? You know, 50% only they will get the success. So 50% is loss. Loss. Okay. So here the 50% again what happened? 50% well growth seed. Well grown. Okay. Well growth seed. Okay. Taken from this one and also grow up time. Next time. The second step. Second practice. This is the first practice. Second process. Here the 75%. Next 80%. 89%. 99%. When it attained the 99%, that seeds are collected and also collected and approach the farmer and introduce the farmer. That is very, very important, my dear students. So we have to practice it. How many times is there? No, 50 or 100 times. This will take up to which one? You know, 50% to up to 100%. It will take more than 100 times. Sometimes it may be exceed. That is very, very important. Okay. Okay. So listen here, mate. So we have to practice. After the completion of practice, we may come to the conclusion. So this seed is very good. So you will attain 100% result. Okay, when we will obtain 100% result, that seed also forwarded to our introduced to the farmer and also in market. So this is very, very important. Suppose if you will not get up to more than 75%, means what is that? That seed is not good. Okay, the maximum obtain this much only, you know. So it is not a good result, isn't it, my dear? That is the things. See this one. Okay, go to the next one. Plant introduction. Next heading is plant introduction. How it will be introduced. See that when a variety of lions means what is the meaning of lions? Nothing but a plant or a population of a plant species is taken from one area into a new area. That is also very, very important. So for example, taken examples of apple. Okay. Apple only grow in the hills place, isn't it, my dear? Hills. Here you have to take different skill station in the India. Okay. And also, same process also done other countries also. Comparison. That is also new area, one area to into new area. Where it is what? Where it was not grown before. It is called a plant introduction or simply introductions. For example, take an example, cabbage or carrot only they will grow in the highest place, highest latitude. Take an example, Kodaikanal, Ooty, and so on. So nowadays, what happened? They will grow in the normal ground also. They are creating the atmosphere or that habitat similar to that of the which one hills places. Then only the plant will be grow. Cabbage. Okay. Take an example, plums. Some of the varieties. Okay. See that. Such introductions have given us many of our valuable crops such as potato, tomato, cauliflower, and grapes, guava, etc. Soya beans is an example of a recently introduced crop. Soya beans, they only grow in the hills place only. Nowadays, what happened? The soya beans, they can grow in the normal tropical region also, tropical. Okay, my dear. So this is also very, very important. This is also very, very important. So plant introduction, where it will be introduced, where it will not grow. We can introduce in the variety and cultivate the plant also. You will get the yield 100%. That is also very, very important, my dear. It is two types of crop introductions. How it will be? That is very, very important, my dear. So many times they were asked the question in this part. Don't forget. IC, indigenous collection. So first of all, you should note it on IC, indigenous collection. Second one is exotic collection. These are the two types. Indigenous. What is my within? See here. When the introduction is made from the same country, suppose if it is the same country, it is called as indigenous collection. For example, Sonalika. Sonalika, what is the name of things? Sonalika is nothing but the wheat. IRS, IR8, IR24. I told it's a rice. These are the two varieties rice. These are all are present in India. Why? Indian rice. Sonalika is a wheat. Sometimes they may ask the question, the name of the plant. Name of the plant also. Okay, my dear. So this is very, very important. So don't forget, my dear. And the next one is to introduce then introduction is made from the foreign countries. For example, Taichung native 1 is one of the rice. See here, Taichung is from China, I think. Okay, rice. Sonara 63, Sonara 64, Lemma, Roja 64. Yeah, wheat. These are all our wheat variety, but this one is also here. Which one you know? Here Sonara, here Sonalika. Know the difference. You know the difference between these two. This one, Sonalika, within the country. For example, India. Okay. Suppose if it is other country, means they have given us different names. See that exotic. Taichung, native 1. So Taichung, native 2. It may be different names also. 1, 2, 3, 4, 5. Like that it may. Rice, Sonara 63, Sonara 64, Lerma, Roja 64, wheat. These are the difference. You must note it down here. Two things is very, very important. One is called as, what is that? No? Indigenous means within the country. Within the country. Same country. That is very, very important. Okay. Here, other country. Know the difference. Okay, my dear. So introduced plant may be used direct for cultivation. That is called as a primary introduction. Or may be used after subjecting it to selection, hybridization is a secondary introduction. First of all, directly, for example, brinjal is there. Okay, just imagine brinjal or mustard or castor, whatever it may be. Take an examples of potato, tomato, Okay, whatever it may be, any one of the plant directly introduced from the country, first to second. Sometimes what happened, the first one selected from the country and also it improvised. That is called a hybridization process. First one is hybridization. Finish it. Some modification also done. Then it will cultivate. That is called a secondary introduction. First introduction means direct cultivation. Second introduction means what is that? May be used after subjecting it to selection process. That is very, very subjecting to selection or hybridization. Hybridization is nothing but the addition or deletion. That is very, very, but secondary introduction is my idea. For the next one. Here, one of the example has given. One of the example. What is that main, Argimon mexicana is an example. What is that? Argimon. Okay, media. Argimon mexicana is an example of weed that enter India with an introductions. What is the meaning of introductions? Unexpectedly, it come to directly from the other country. Okay, from the other country, from other country to India, like that. Imagine. Okay, therefore, all introductions are carefully examined for the presence of weeds, insects, and disease-causing organism. This is known as, what is that? Quarantine. This is called as quarantine. Okay, already we are in the quarantine period. 2019, 20, 21, isn't it, my dear? Quarantine means, what is the meaning of quarantine? See here, once again, is an example of weed that we enter India within the introduction. Therefore, all introductions are carefully examined. That is called as quarantine. Examine means it is under the research. Okay, so that process is called as quarantine. Not yet introduced. For example, COVID vaccination, not yet fully discovered, isn't it, my dear? So, before that year, when it entered in our country, so that stage is called as, what is that, quarantine stage. Only those introductions that are free from the above or what is it, permitted to be used and the rest are destroyed. Okay, so what does it mean? We are introducing a new variety, isn't it? New variety. Just take an examples. Okay, that's just take an example. AB1 is a COVID-1. Just imagine. COVID-19. Okay, just imagine. This is invading. After that, what happened is there are so many changes occur, isn't it? So many changes. Last one is also there. Okay. How many changes is there? No, hybridization. Okay. And also the modification of gene level, isn't it? You will get new variety. Sorry. New variety. Isn't it, my dear? So, when it enter from the China to India, we do not know about the COVID virus, isn't it? Virus, physical process and chemical properties, whatever it may be, we do not know. That time, this virus is under the research in laboratory in Pune, particularly Pune. Okay. Virus under the research. That condition is called as quarantine. After that, what happened? It may be modified in some other form. Delta, so many things. Okay, modification is there. So the first one is, what is that? Deleted one. New variety only is available, isn't it, my dear? So this is the consider. This is the consider one. Remaining things completely, what is that? Destroyed. Not only destroyed, completely omitted. Inavailable, omega. Isn't it? So many things is there. Okay, recently. See, this is the man. See that? Only those introductions that are free from the above are permitted to be used and the rest are destroyed. Okay, likewise, which one is introducing? That one is, what is that? Permitted. Which one is permitted one? That species only we were used, otherwise remaining things are completely destroyed. Okay, it will not get the good result. That is the reason. It is not, it is also related to which one in a plant as well as animals also. Okay, what the next one? And then wheat, an example has given. One is a dwarfing gene. Dwarfing means it's a short name. Short, tall, no, it's a dwarf one. The variety is called as, what is that? The wheat is the one of the example has given, wheat variety. Okay, the wheat variety particularly note it down. So the Sarpana, Sonara, all those is Sonalika, all those things already we studied, isn't it? Here what they given, dwarfing gene of the wheat, Norin 10, Norin. See, this 10 was reported in Japan. See that Japanese. Norman E. Porlock is the name of the scientist. Just imagine Norman E. Porlock is the, which one, Green Revolution, the World Green Revolution. Okay, given by the name of the scientist Norman E. Porlock. At the International Center of Wheat and Maize Improvement in Mexico, they have given us a CMMMYT. So what is the expansion of this one? Sometimes they may ask the International Center of Wheat and Maize Improvement in Mexico. What is a semi-dwarf wheat? That is very, very important. Semi-dwarf wheat. Already told you. MS Swaminathan has also done the one. Sonalika. Sarpana Sonara. Like that. It's also one of the wheat variety introduced by, discovered by the name of the scientist called as, what is it? MS Swaminathan in the year 1966. So this variety was discovered in which area, Mexico. That's why they have given as a name is called as popularly known as Mexican variety. Which one? Norin 10 was named as a Mexican wheat. Who stated the statement? Norman E. Porlock. This had high yield, resistant for lodging and common pathogen. What is mean lodging? Storaging process. We can store up to 6 months, 10 months or 1 year, 2 years, 10 years, like that lodging. And common pathogen pests and smaller growth period. That is very, very important. Smaller growth means short period you will get the yield. That is very, very important. And the next one is 1963, several varieties such as Sonalika and Kalyan and also Sona, which were high yielding and disease resistant. This is very, very important in the each and every plant or animal, whatever it may be. This is the most important aim, my dear. This is very, very important. Don't forget. You can put 100 important. What is that? Very high yield and disease resistance were introduced all over the wheat growing belt of India. So this is very, very important. Which variety? It was discovered in which area? How it will be transferred to here? It is also from exotic species. Exotic. From the outer country to our country. Already we discussed. Okay. Which variety? Which variety? That is called as, which one Sonalika. Sonalika is nothing but the wheat. Just imagine which one of the following is not a wheat. Sometimes they may ask the question in NEET. So NEET question, don't forget. Okay. So he is also a World Green Revolution. Norman E. Porlock. This is the NEET question. Sometimes they may ask the question CIMMYT NEET. It's a expansion. Okay. Sometimes they may ask the question, Narian 10 is also called as which one, Mexican wheats. The Mexican wheat variety, it can be look like which one is semi-dwarf, dwarf or tall or like that. They may ask the question, the properties of the wheat. Semi-dwarf. Which year? Okay, all those things. This is also very, very important. Sonalika is a which type of variety? Whether it may be a dwarf variety or semi-dwarf variety, whether tall variety or high, very tall variety. Like that, they may ask the questions. Okay. So this is very, very important. So don't forget, my dear, go through it. Next one is rice is also given as an example. Rice. Okay. A dwarfing gene. See here the dwarfing gene of rice, DEEGEWOWOGEN. See that rice, what is the name is called as? This was name of the rice was reported in Taiwan. It's a other country, Taiwan, which is the rice bowl of India, Tanjavur, isn't it? So likewise, here what happened? Here the rice variety is also introduced. The name is called as DGO-WU-GEN. DGO-WU-GEN was reported in Taiwan. It was introduced by the IRRI, International Rice Research Institute, Philippines. Don't forget, my dear student. This is also very, very important. Sometimes they may ask the questions. This dwarf gene rice was introduced by the name of the country, like that they may ask questions. Taiwan. What is the expansion of, what is that? IRRI. That is also International Rice Research Institute. Where it located, that is very, very important. So many times they were asked the question. Okay, this is very, very important, my dear. 8 questions. Here, what is that? IR8, IR24, and Taichung Native 1, variety of rice. The derivatives were introduced in 1960s. I told you, 1966 is the, which one, Green Revolution period, Green Revolution. Revolution, okay. Period. Okay, media. So this is the, I think, in the 5th 5-year plan, I think. 5th to 6th 5-year plan. 5-year plan. Self-satisfied, it is also very, very important. Green Revolution done by the MS Swaminathan. Okay, Sonalika. Sarpadi Sonara is a wheat. Sarpadi Sonara. Don't forget, my dear. In the year 1966. Same process. Later, better yielding semi-dwarf variety Jaya and Ratna. Don't forget, my dear, this is very, very important. Were developed in India. So, just to go to the question, 2019 or 20. Okay, these two is there. Jaya and Ratna is one of the semi-dwarf variety, like that they ask the questions. NEET. Very, very important in this part. Rice is very, very important. And see here, G.S. Kush is considered the father of miracle rice. Don't forget. Who is the name of the scientist? The name of the G.S. Kush. Just imagine Kushpu, like that. Imagine. Kush is considered the father of miracle rice. IR 36 developed by crossing 13 variety of rice within India. Rice variety Varaiza Nivera found in Kerala. Varaiza Nivera found in Kerala. See here. It is also one of the variety. Okay, my dear. You must note it down. Rice is very, very important compared to the wheat, particularly wheat. So, Sarpadi Sonara, Sonalika, different varieties also there. And also here the rice is also semi-dwarf varieties. The wheat is also a dwarfing gene. Okay. It is also semi-dwarf. Don't forget, my dear. Both are semi-dwarf only. Okay. The dwarfing gene of rice, DGO-WU gene. This is also very, very important. Gene, dwarfing gene of rice. This is a gene. They may ask the question. This gene is also isolated from where? Reported from where? Like that they may ask the question. Taiwan. Okay. Taiwan. Don't forget, my dear. This is most important. Most important. Okay. So this is also neat. And next one is what is my IRRI, International Rice Research Institute, Philippines. They asked so many times, 2 to 3 times they asked it. So variety is what is that? IR8, IR24, and Taichung Native 1. The Taichung Native 1 is also Taiwan. Taichung. Variety of the derived were introduced in the year 1966. Year also very, very important. This is also important one. 5 questions here in this part. And who introduced that? Father of miracle rices. So the G.S. Kush. How many varieties there? 36 varieties. 13 varieties from India. Total how much is that? 36 varieties there. 13 varieties from where India. The variety is called as Varisa nivera. In our rice, Varisa sativa, isn't it, my dear? But here, Varisa nivera found in the Kerala. And next, hybridization. Okay, hybridization, how it will be? Hybridization is the most common method of creating genetic variations. Creating genetic variations. A line consists of group of individuals released by decent and often similar of genotype. We are selecting the rice or wheat, whatever it may be. Which one it possesses, rice or wheat possesses, which one, similar genotype. Similar. That is very, very important. Similar genotype. Then only the hybridization possible. Suppose if it is not similar, means what happens? The hybridization is not possible. The individual lines used in hybridizations are called as, what is that? Parents. The mating between two or more individuals or line differing in the genotype is called as hybridization. Differing. Suppose if it is differ. So that is called as hybridization. Such mating constitute intervarietal hybridization. Intervarietal. Intervarietal means different. Intravarietal means same. Inter means what is a different gene sequence. Different. Just imagine. Intra means within. This different, same variety. Okay, my dear. So these are the very, very important factors. So the next. But sometimes a crab species is matted with the different but related species. This is known as interspecific hybridization. Interspecific hybridization. What is my interspecific hybridization? Just imagine sometimes a crab species is matted with the different but related species. Different but related species only. Such type of hybridization is called as interspecific hybridization. The interspecific hybrids are ordinarily difficult to produce as well as use, but they are important in plant breeding, particularly in breeding for disease resistance. So particularly for what is a disease resistance. So interspecific hybridization mostly important for plant breeding, particularly disease resistant process only. Some interspecific hybrids are even used as a variety. For example, all the sugarcane variety being cultivated today are interspecific hybrids. So take an example, interspecific varieties. Which one of the example? It is also very, very important. NEET. Here also you must note it down. NEET, important. So two things is you must note it down. Interspecific is just a disease resistant only. And also, which is the best examples, means sugarcane is the example. Saccharum officinarum. Saccharum officinarum is an example. It's a botanical name of sugarcane. Okay, interspecific. Within the, which one crab species is matter with a different but related species. Related species. See that one. Polyploidy. Next one will pass on to the next one is called as another subheading is called as polyploidy. What is by polyploidy? The polyploidy species individual are the following types: autopolyploidy, allopolyploidy. Already we discussed. Autopolyploidy means what is that? Auto. So spontaneously they produce it. In naturally. See that autopolyploidy is received spontaneously in nature. Spontaneously in nature. That is called as autopolyploidy. In low frequency, it can be induced in relative high frequency by colchicine. Don't forget, my dear student, colchicine is also one of the chemical substances. Chemical substances used for the genetic engineering. Already we discussed. Particularly, what is the purpose of this one? Autopolyploidy. The colchicine is an alkaloid, the chemical substance I told here, the alkaloid substances that prevent the formation of spindle apparatus during mitosis. Don't forget, my dear students. This is also mostly used for the cancer divisions, cancer mitotic divisions. I told. Okay, the colchicine is also one of the very, very important chemical substances. Particularly, they told the alkaloid substances present in the some varieties. Okay, my dear, some varieties. Okay, my dear. So you must note it on this point. Sometimes an autopolyploidy has succeeded as a variety. For example, autoploidy variety of tea in India. So one is called as sugar. What is that? It completely prevent the mitotic spindle formation. The variety, particularly tea, is an example has given. Most of the present day crops such as tomato, grapes, and watermelon, banana, pineapple, or autopolyploidy. Maximum examples they asked. Which one of the following is a autopolyploidy? Which one of the example is not related to the autopolyploidy? Like that, they may ask the question, my dear students. Okay, my dear. Today, so we will finish it up to this level only. We will see the remaining things on the next class, my dear students. Okay, I will summarize all those things within short period. What are the things already we discussed here? First of all, plant breeding. What are the steps is there? You note it down. Crop genotype, animal genotype, environmental under which the crops and animals are raised. The things already we discussed. Development of the new variety. What are the things is there? Creation of genetic variation, selection, evolution, and release as a variety, seed multiplication, and distribution on farmers. These are the steps is very, very important. And then genetic variation, how it will be created. Already we discussed domestication, germplasm collection, plant introduction, hybridization, polyploidy, mutation, genetic engineering. Each one it has a specific properties and things. Already we discussed all those things. Domestication, germplasm collection, okay, and plant introductions. And what are the two types? IC and EC, very, very important, my dear students. And which one of the example has given? Okay, what is by quarantine? What are the examples of wheat, semi-dwarf wheat? What is by CIMMYT? And Sonalika, when it will be introduced in 1963? Kalyan, all those things. These are all a semi-dwarf variety of wheat. And rice, what is that? Taiwan. What is the gene name is called a DGO, Wu gene. Okay, IRRI, what is that? International Rice Research Institute, Philippines. Okay, my dear. And then IR8, IR24, types of native, all those things. And 1966, they were introduced the new one, Jaya and Ratna. And these are the Jaya and Ratna. Don't forget, my dear. This is very, very important. GS Kuss and father of the miracle rice, IR38, developed by crossing 13 variety of rice within India. Rice variety, Varisa Nivora, found in Kerala. Okay, what is by hybridization? Okay, which one is the parent one? Okay, interspecific hybridization. What is that? How it will be? Particularly two things is very, very important. It considered as two things. What is that? Disease resistant, and which one is the best examples of this one? Sugar cane. And what is by polyploidy? Already know about polyploidy is nothing but the species individual or the following type. Autopolyploidy. Automatically means natural modification. Autopolyploidy is produced spontaneously in nature in low frequency. It can be introduced in the relatively high frequency by colchicine. Okay, colchicine is nothing but the alkaloids that prevent the spindle formation. Okay, my dear. So remaining things we will see the next class, my dear students. Thank you so much for the participation. Bye. Good night. Thank you, ma'am.
Thank you, sir.
Thank you.